I thought I'd say a few words about this new research center and then hand over to Fabien to talk about the research going around the FoxG1 project with your funding. And this time without a whiteboard, so I even have a few slides. Huh? So just for those who don't know me, I'm Lars Sittner, I'm the director of this center. We came to Macquarie University end of last year in December, and have been, welcomed with open arms and found an extremely supportive environment here. So our research over many years has focused on neurodegenerative diseases, developmental diseases, as well as age-related dementia and associated conditions. And we had I would say, a very successful research unit at the University of New South Wales, but within the constraints of that unit, it became clear in the past few years, if we want to really take the next step to develop our discovery research into therapies, we need to find an environment that is really truly supportive of neuroscience, and it is needless to say that Macquarie is such an environment. So the Dementia Research Unit changed fundamentally by becoming the Dementia Research Center, and we obviously moved to Macquarie University. So the center in a nutshell has 4 main tiers that we are researching or developing. The first one is we continue to advance our discovery research program. We develop preclinical models, and this is where Fabien is a centerpiece to what we are achieving. And we have started a program that has now embraced 14 different drug targets to develop new drugs for different neurological conditions. They span from epileptic conditions over rapidly progressing muscular degeneration to obviously dementia. And a very important aspect that we particularly in the past few weeks, with a lot of students coming through the center, are building up is the education and development of new leadership for the next generation. I always say the next generation is the one that is going to solve the problem. I hope my generation is going to understand the problem eventually. So, don't have to talk about myself. So Thomas is our deputy director and has taken the portfolio of education under his wings. He is an extremely experienced teacher in addition to his research achievement, and he's running our cellular neurobiology program. Jadze is the deputy director operations, who's looking after the overall operations of the center and is driving a research program focusing on gene therapies, which has become in the past few years extremely exciting to work in this area. And this is the good heart of our center, our manager, Amy Law, who's really keeping everyone in line. You all have met Fabien. He's going to say a few words. He's heading here at the genome editing service at Macquarie, which is soon to be incorporated into our center, and he's the driver behind disease modeling and in animal models. Arne, which is I think you can see by resemblance, is also my brother, so he's my longest term collaborator. He is driving a research. I'm extremely proud to be working with my brother. He's driving a disease mechanism discovery research program. Janet has really grown under my wings from a junior student to now a really active group leader, and she is driving together with me the drug development arm of the center. And finally, we have reached out into other disciplines and have recruited Adam Martin, who is a chemist by nature, and it's really opening up our horizon in things that are doable, which I've never dreamt of. So this is the group that actually came over from UNSW with a few new recruitments, and we are now hitting soon the 40 people. So the group is ever growing. You know, if you see the faces around here, approach them if you want to know what they're doing. So they're all very approachable people. Our center is internationally connected, so these are just an outline of our active collaborations. And this is not just collaboration where someone sent us a reagent. This is really researchers we engage on a daily or weekly basis in our research program with. And if you want to learn more about the center, then visit our homepage. Here the link is at the top, and you find information on dementia as well as on our research programs and the people working here. And you have opportunities to see how you can get involved if you want to. And finally, the modern days have kept up with me, so that's something I'm not familiar with. So this is the more junior people running these, as is a Twitter account to keep actually up on what's happening at the center. And I think that's enough from me. We're really here to hear about our FoxG1 program, which I think within the scope of the Dementia Research Center, which is focusing on the big problems of Alzheimer's disease and the like. It is these projects on very particular questions and conditions that really spice up our life and make it really worth coming every day in and doing the job, right? Okay, so I pass on and we learn more about FoxG1 from Fabien.
All right. Okay, so thank you very much, Lars. Before I start talking about the journey as I lived it from the beginning, I just would like to acknowledge what Lars just said. What he just showed you is something quite exceptional. The environment in which we work all together is something very special. And I believe the only reason why we're here together is that because, as I mentioned to you, Vivek, in quite of the few fundraising that we did, this is only possible when everyone comes together. So I think if I tell you the story the way I lived it, it started like that. One day I received a phone call on my personal mobile phone. And I'm still to work out how you found my personal phone number, so you're gonna have to explain me that one. And the reality is I usually never pick up a phone number that I actually don't know. And for whatever reason, this one, I did pick it. And that was a good idea because this is not the kind of call you hear on a daily basis. This was someone who already started to try to establish a roadmap for a cure for his son. He tried to establish that by putting people together, and unfortunately, it was quite unsuccessful. I believe Vivek contacted people and researchers in America, and for many, many different reasons, including geographic distance, etc., the project didn't actually fall out together and in place. So I attentively listened to what he was saying, and what was really striking is that I was somehow, as Lars mentioned, at the right place at the right time, because when a program has to start on a rare genetic disorder, obviously, on a scientific point of view, the first thing to start is to try to develop cell and animal models of the disorder. And that's precisely what I've been doing for the last 15 years. So the reality is, as much as I was excited for this project, I had to talk to Lars, because without the support of the Dementia Research Center, as you've seen it, and the faculty as well, we wouldn't be there. So I think what's really important, as I mentioned as well last time, is these sort of project only are achieved only when everyone comes together. So your part, the fundraising part and the community, the support with the donations, the banks, etc., would go nowhere. And we, on our side, the scientific part, would go nowhere neither if we were not coming together. And I believe this is one of the exceptional example where we finally, by coming together, made it. And for those who don't know, Daniel is the recipient of the FoxG1 PhD scholarship, which after a few meetings, obviously Vivek needed a bit of guidance on how to actually set up such a project. We decided to set up a scholarship, and with Lars' help, we actually had the right person and the right place to start this research project. So now I believe, Daniel, you are on your second year of PhD. We have successfully finally generated the cell models and as well as the animal models of the conditions. So we replicated the exact same mutation that Kush carries. And the idea now is to try to understand exactly how the mechanisms that underpin the disorder. And Lars mentioned as well that we are lucky enough to have a gene therapy unit here that is led by Yazi. So obviously, as you can imagine, when you pinpoint the problem to one specific mutation, the idea is you can also use this gene therapy to try and rescue the problem to a certain extent. So that's also the final goal and what we are trying to achieve. And that, as Lars mentioned, has really a translational potential because it goes way beyond one specific disorder. So I think that's also a reason why we're trying at the moment to incorporate the FoxG1 program into a bigger program that has to do with seizure-related disorders. And I think we've achieved quite a lot. If you want to talk about the very specific of the scientific ideas that we're trying to develop, we can do that after. I'm gonna give you one indication only here. If you have heard about mitochondria, which is a subcellular compartment of the cells, we tend to orientate the research into this direction. So we try to obviously test and gauge different approaches, but this one seems to be realistically the one we're going towards. So it is a very exciting part of the project, one that now materializes with what Daniel is doing on a daily basis. And hopefully it will translate on a global space. So once again, I would like to acknowledge first the FoxG1, the Australian FoxG1 Foundation. Without you, Vivek, without you, Monica, without the kids and without the family, the community, we wouldn't be here, we wouldn't be doing this project. And without the support, as I said, we would not do it neither. I hope we're going to continue and grow up, both in terms of scientific project, but also in raising awareness for the community, because we had a meeting just an hour ago about this. This is the way to go. And the more people get involved, the better the outcome. So thank you very much. Thank you, Amy, for organizing as well. Thank you, Josh. And thank you everyone for coming. If you have questions, we'll be happy to answer very specific questions, but now I'm just gonna maybe give you the opportunity to tell you the story on the family part, so that everyone knows exactly on the personal, very personal basis what this project is about.
So, could we ask, like, if all stars align, what's the expected time frame to move from primary to clinical to?
So from primary to clinical, I can't give you a time frame. From primary to the end of preclinical, that is realistically doable. This is what we do. as we discussed many times, the translation to clinics, and that's why I'm saying we need to even bring a bigger community, including scientists who are clinicians and who will tell us, because this is not what I specialize in, what the next step will be and how long it's going to take. So as much as I would like to give you a time frame, I'm not able to do that, but obviously, as you can imagine, everyone is doing their best to make a time frame that is as short as possible.
Should I share it?
Yes, absolutely.
Certainly, wanted to thank Lars and Fabian. Without you, like, we wouldn't have had any scope or any chance in at all. But also Josh and the directors, like I think Amna, like we met at a COC part, and like Amna jumped in and like, she's been trying to help Ellie and people from Ellie's a third-year med student, and like she's got so many people running for us. And I think while we are still a very small charity, I think if you go back, I think the way I understood the condition was that that blue line needs to be turned into an orange line. That's the only thing we're trying to do, and that's where you are the experts at. So he's got one small mutation. We fix it. It should hopefully, or like we look at the options and like if the mitochondrial option would be the best. When we started, certainly like I was speaking to, and there's a lot of people who bring us to this stage where we are able to do things that we're doing. I think Sun was the first person I spoke to who actually said that Sun Sunte was a an oncology researcher at LAi, and so she mentioned that like, oh, if you need to get anywhere, you need to find an animal model. So we said, hey, who's animal model? So Fabian's PhD is on Sydney Uni, and so I said animal model PhD, and so Fabian's name popped up. That's how I called him up, and like there was, I think, and having even Daniel work for us, like we just doing a very small part of his research. We are supporting a very small part of the research project because like the whole infrastructure, the whole hospital, everything is needed to make it happen. So we're so grateful to get all of the help from everyone. But I think I also wanted to just specifically share one story about Lars, because early on when I met you, we were having a discussion and you mentioned to me that a story about your life where you were doing a certain experiment, that you were doing it every day for 6 months. And you wanted an expected result, and like it didn't work out. Second day you kept coming, and it didn't work out. Third day it wouldn't work out. So after 6 months, like you got it worked out. The experiment worked out properly, and then you said that like the tenacity behind some of these things it's not an easy story. If it was easy, like it would have been done. It's while it's getting easier with technology and everything, I think certainly in the last 3 years as well, I think in terms of SMA and like the amount of gene therapies that are happening, and like in China, the AIDS-free babies and things, I think ethics is one of the lesser. We were also like, if today we had a cure, would we be able to administer it? Would have been a question 3 years back, but now maybe potentially that's a lesser question. So I think previously it was, will it happen or can it happen? But now it is, how much will it cost to make it happen? And I think the costs are also coming down. And like, again, through, I think our belief that this will happen, this will work out. We had a very honest discussion with Fabian whether we'll get to the 100% answer or not. But I think as a result of this, we'll at least get to a step ahead of where we were before. So even that's a great journey. And I think I truly believe that's like without everybody's help, this wouldn't have been possible. There has never been a better time than now to make it happen. And I think we are so lucky to be in this country as well, because even from the government as well, there's a lot of good things that are happening. For a small baby who died at the age of 7 months with SMA, so now there was 500 million that was awarded towards a future genome research fund. So we are so grateful. Like I, on behalf of like everybody, is so grateful to all of you superstars for making it happen for us as well. Thank you.
Thank you, LAi. Thank you. I still don't know why the experiment worked after 6 months.
I just thought it was nice that you're not Superman after all. You can't do everything. Do you want to speak first? No, go for it. Happy to. Fire away.
Can I stay here? Is that okay? Yeah. So I just wanted to share the grant that we provided last year. So I'm the branch manager at the Commonwealth Bank in Stanhope Gardens. So last year, every branch across Australia was given $10,000 to provide to a charity of their choice. And I didn't want to make that decision myself, so my team and I came together. And I think it was one of my staff that knows Monica outside of the community. So she brought that to me by the end of the day. I went home and did some research and, read about it online and Facebook. And to be honest, it was a really easy decision. We stopped looking after that and invited them in for a morning tea and presented you guys the cheque. And I can see the passion. You guys said to me that day, you're not going to stop until Kush can run around with his brothers and sisters. And I feel really honoured to be here and hopefully be a part of that. So thank you.
Do you want me to stand up or can I sit down as well? Oh, what? Okay. I don't normally talk, I normally delegate speaking duties, so I'll have to make an effort now. So look, my role here is actually to represent the university. So my role is as the Associate Dean Research in the Faculty of Medicine and Health Sciences. So effectively I'm responsible for all of the research that we undertake in the medical precinct. And so it is in that role, on behalf of the university, I'm very pleased and excited to welcome you all here and to thank you for your contribution. So Kush, especially, and your family, so Vivek and Monica and kids, and our other guests, thank you very much for your support of this project. I think you've heard how incredibly important that is for actually making this project occur. Lars wanted me to talk a little bit about the university and I guess in some ways why they've chosen to relocate here. So you may be familiar a little bit with Macquarie University. We're unusual in the fact that we have our own hospital. So about 10 years ago, the university invested in building its own hospital. We've also now built a medical school that links to that hospital. And so we're the only university in the country that has an integrated academic health science centre where the hospital, the medical program and the research and education is all actually under one umbrella. And that's particularly important as we think about what we've heard today, because if you want to actually integrate and deliver translation, having that actually in one place is really quite important. You spoke, Vivek, about some of the regulatory challenges. So for a very simple example, if you're working in another university, in another hospital, you need to have separate approvals for your projects through those different bodies. We have one single process, and so that makes that streamlined. It's a lot easier. The mission of our Academic Health Science Centre, we call it MQ Health, and really we have three goals. Heal, learn and discover. It's really quite simple to remember, and really that encompasses what we're trying to achieve. But the value I wanted to talk to you about today, the values that we have for the organisation, one of those is around caring. And I think that's really relevant here, because if you look at how this is happening and the way the conversation is going, you can actually feel that there's actually a lot of care for this project. And that's something that's true with our Academic Health Science Centre. So we have a number of patients that come through the hospital and through the clinics. And we're particularly interested in bringing those patients in to contribute to our research and to our teaching. So as a simple example, I work in the motor neuron disease research program. We have the largest motor neuron disease clinic in the country. So we see 10% of all the patients in the country come through our clinic. And every one of them contributes to our research. Every single one of them. So we meet them, we engage them in our project, we show them around our labs, because it's really important. We actually care about our patients. And I think that's true here, and you've heard this from the team here. And I guess that's how I wanted to end today, by really welcoming you to our family, the Macquarie family. I hope that you'll actually visit often. I expect Kush to be coming in to supervise these guys, because we need Lars and Fabian to work harder, and so they need a bit of a push. Yazi's not enough, right? But you're part of our family now, and I hope you'll actually feel that, and I hope over time as we get to know each other better, you'll actually recognise that. It's great to have you all here. Thank you very much for your generous support of the program to keep it going, because I know it's moved from UNSW, and I really see that this is just the start of a much longer and hopefully a journey that will get us to where we want to get to. So thank you very much for all of you today for joining us.
explaining everything that has to do with cells or molecular level is happening here. And whatever we find interesting, we try to translate that directly into the mice. And eventually this, if successful, will be translated just next door. But it all really starts here. When you have a problem such as a genetic mutation, this is the perfect environment to test whatever hypothesis. Not only can you replicate the exact same mutation, but you can test it in different settings, because what would work in a dish would not necessarily work and translate in animals, and vice versa. And there's many different assays that we apply in this sort of setup. And what Daniel does, because I do nothing, is on a daily basis he works here, he does all these investigations, and then on a weekly basis we do a kind of meeting where we decide what happened, did we face any technical issue, and how can we overcome that, because quite often the solution comes from technical limitations rather than ideas. We have quite a fair bit of ideas. The reality is, how do we actually materialize the ideas? And for that, I don't know if this is the first time you enter a lab, but we have a lot of very fancy equipment which look like we're going to go on the moon within the next hour or so. The reality is, when you're a researcher, I guess, whatever your job, you have some pieces of equipment or buildings or places or techniques or anything that you do, whatever the process, which is very specific. And if I come from the outside, I would probably find it really amazing. But we work with this kind of equipment on a daily basis, and the reality is, as fancy as they look, what really makes a difference is what can they actually do, and how do you understand, how do you make it work. It's like having a really fast car. If I really don't understand how the car works, or if I'm not really trained at driving it, I'm never going to make it going fast. So these are very, very expensive and specific equipment, but without that we would never be able to explore projects like FoxG1. And we're going to walk all around, and I'm going to just show you these pieces of equipment. If it doesn't talk to you, just ask me, what is it for, what do we use it for? It may be much more relevant than telling you this is this or this is that. Okay? The first lab, which is a very specialized part of the procedure, is just behind you. And the machines that you see there, so the big ones are called centrifuge, but the ones at the bottom are what is called Pcr machines. And these machines that are blinking in green here are the ones that they do tell us, they're able to give us really cues as to what the genetic is behind. So if we have a cell that has a genetic modification such as the Kush mutation, using these small machines here, we can, for instance, identify whether this one mouse carries the mutation and this one doesn't. So by kind of a recipe, this is nothing else but cooking, you just need to know what you're actually cooking. These small machines here will tell us whether the mutations are present, and also if we can find a way of reversing the mutations. So these are called Pcr machines. The only limitation in this one is very interesting kind of piece of equipment. The only problem is to visualize the DNA, we need to use a cytotoxic kind of dye, which is why I'm not going to let you in this one, but it is quite of a heavily used room. As you can see, the work plan is an open plan, which is really great because we can share our ideas and work together, particularly when the experiment starts at 7 and finishes at 11 in the best case scenario. So it's really nice because you can actually interact with your colleagues. all the chemicals, etc., needs to be stored in special places, and they need to be discriminated. So here, for instance, you have corrosive substances which you can't mix with flammable, because if you do, then you blow out the building. So we have to have people who are familiar with these sort of things on a daily basis as well. So when I say research is about knowing what you do, these sort of considerations apply as well. Here is dedicated to the production of viruses. So as you can imagine, once again, we have very stringent regulations, because these viruses, Vivek, you were saying, if we can simply reverse this mutation that was on the banner, then we would simply fix the problem. I wish it would be that easy, but the way you bring the medicine to the cells makes it another challenge, and that's where we kind of overcome the challenge. For that, we use viruses. The drawback is if we can actually develop a virus that is very efficient in penetrating the cells to correct the mutation, it will also be very efficient at penetrating your very own cells for any other reason. So we got to be very careful with that. So the virus culture is happening here, and the cell culture, which is where, for instance, we develop induced pluripotent stem cell, is all happening here. So that's why we discriminate the two workflows. This is absolutely essential for the FoxG1 project, because you can test hypotheses really quickly on cell culture and have an answer to whatever hypothesis, and the readouts, which is quite pretty much instantly.